Good morning, everyone. Thank you. Welcome to our annual IMS Scientific Day. I'm Cindy Morsehead. I'm a graduate coordinator and chair of the organizing committee for our event this year, and we're all thrilled that you could join us today. Each year, Scientific Day offers the IMS an opportunity to celebrate the achievements and the excellence of all of our faculty and our students. We truly are an institute that crosses and engages both the basic sciences as well as the clinical sciences across many research themes, across the hospitals, the research institutes, and the campus, the University of Toronto. We're one of the biggest and most diverse graduate programs in the Faculty of Medicine, so in fact, we have a lot to celebrate. This year, we put together an exciting agenda of what we've called firsts. Hopefully, this will allow us to highlight our research excellence. It's going to provide a venue for us to foster collaboration and celebrate this diversity. We have a number of new initiatives to make this happen. First, this afternoon, we have 3 concurrent breakout sessions. I'm particularly excited about this. We have 3 exciting faculty from the IMS giving talks, which will be followed by students highlighting in 5 minutes their data blitz about their current research. We also have a new speakers corner. This will give people an opportunity to give a short interview about their research, and that's going to happen right in the grand ballroom over the lunchtime period. For the first time at Scientific Day, we also want to extend a warm welcome to our prospective students, students thinking of joining the IMS to do their graduate studies. We hope you enjoy the day, learn all about the exciting research that's being done, and learn about the opportunities that are afforded to you at the Institute of Medical Science. We look forward to seeing you at the Lunch and Learn that will be held over the lunch period. We've already gotten off to a great kickstart. We had our poster competition, our Alan Wu poster competition this morning. We'll be hearing about the winners later this afternoon. I hope the rest of the day provides the events that will maintain a lively, engaged, and enlightened audience. At this time, I'd like to invite Dr. Mingyao Liu to come to the stage. Dr. Mingyao Liu will introduce our keynote speaker today. He is the director of the Institute of Medical Science, an MD, and a professor in the departments of surgery, medicine, and physiology. He has a very active research program examining the cellular and the molecular mechanisms of lung injury. His group also uses state-of-the-art approaches to exploring the role of adapter proteins in signal transduction during lung tissue injury and repair. Dr. Liu has taken on a number of roles in the IMS, and he's been part of the Institute back to the year 2000, and in many capacities. Most recently, in January of 2015, he became the Institute Director. We're thrilled to have him in this leadership role. So please join me in welcoming Dr. Mingyao Liu.
Thank you, Cindy, for such a nice introduction. And also particularly thanks Cindy and the organization committee for the Scientific Day to transform the format of the IMS Scientific Day. I hope the new format will be successful. So before I introduce this year's guest speaker, I would like to give you some background history on the Bernard Langer Annual Lecture in the Health Science. So the lecture was established to honor the achievements of Dr. Bernard Langer in founding the Surgeon Scientist Training Program at the University of Toronto during his tenure as the chair of the Department of Surgery from 1982 to 1992. So each year, a guest speaker is selected who is internationally recognized for his or her scientific contribution. So today, Dr. Langer is with us, and thank you very much. So this year, we are honored to have Dr. Christopher Austin, Director of the National Center for Advancing Translational Science at the NIH, to present the Bernard Langer Keynote address. So Dr. Austin joined NIH in 2002 as the Senior Advisor to the Director for Translational Research at the National Human Genome Research Institute, where he was responsible for conceptualizing and implementing research programs to derive scientific insights and therapeutic benefits from the newly completed Human Genome Project. So while at this institute, he founded and directed the NIH Chemical Genomics Center, the Therapeutics for Rare and Neglected Disease Program, Toxicology in the 21st Century Initiative, and the NIH Center for Translational Therapeutics. Shortly after the creation of NCATS in 2011, he was appointed as director. So Dr. Austin earned his medical degree from Harvard Medical School, and he's trained as a clinician and a geneticist. He trained in internal medicine and neurology at the Massachusetts General Hospital in Boston, and practiced medicine in academic and community hospitals settings, as well as in urban primary care and in rural Alaska and Africa. So please join me in welcoming Dr. Christopher Austin, who will present his talk on the Catalyzing Translational Innovation. Welcome.
Well, it's a real pleasure to be here today, and I love giving talks to students because, as you'll see, I'm going to issue you sort of a call to arms here, so be prepared. I do want to mention, before I get started, I just had a chance to meet Dr. Langer, and it was really a wonderful thing to realize what a mentor you have and what a role model you have in Dr. Langer. We did overlap institutions in the Harvard system, although I must say that he preceded me by 3 decades there, but actually the same people a little bit later, and so I hope you'll all consider his example in your own work of applying scientific principles to improve the health of patients. So what I'm going to do today is to give you a really rapid tour through how we're thinking about this problem at NCATS. And as Dr. Liu said, NCATS is only about 3 years old, so we really are like a toddler, 3 years old. We're out of the terrible twos, but we're still we have a lot of potential, but we still got a lot of growing to do. But also so think of this sort of a work in progress. So I just want to start out with showing you my world in the last 30 years. So I graduated from medical school in 1986. And so this is a graph that goes from 1987 to the present, and it's a graph of the number of disorders in OMIM, and the online inheritance in man, that have a molecular basis. First of all, I got to ask, is this a teaching session? Does anybody know what is the first disease, the molecular basis of which was determined? Sickle cell. Somebody actually knew. This is like the second time ever somebody's. Okay, you get bonus points for knowing the year. 1949 is the year. And who was it? Linus Pauling. Yeah, this is really great. You ought to go on Jeopardy. This is really good. Very few people know that. So 1949, but from then up until about 1987 or 1988, 1989 was cystic fibrosis. 1991 Huntington's disease. So very small, very slow slope. But after that, the number increased dramatically, and the reason it increased dramatically is concurrent with deposition of large amounts of sequence data into public databases as part of the Genome Project that happened. The sequencing phase of the Genome Project started in the mid-90s, and this is when this happened. So the good news is that today, as compared to when I was in training, there are less than 50 disorders, a molecular basis of which was known, actually about 20 at the time. Now it's about 5,500. So for each of you who's starting a lab, each one of these is a project, and each one of them are patients out there in need of your attention. Because the bad part about this is there's only 500 of them, and this is a generous estimate, actually, that have any treatment. And so in my training as a neurologist, most of the patients that I saw had disorders with no treatment. And still this is the case. If you think about what neurologists see, they see ALS, Alzheimer's disease, Huntington's disease, intractable pain, brain tumors. These are essentially, for the large part, untreatable disorders. And it's not just neurology, it's across the field of medicine. And so enormous need and enormous opportunity now that simply did not exist 30 years ago. Okay, so the story of sickle cell is a cautionary tale, though, because here is the paper, the original paper, Linus Pauling, 1949, Sickle Cell Anemia, a Molecular Disease, and there is still no drug directed against this molecular pathogenesis now 66 years later. So just because you have a gene or gene defect doesn't tell you that you're going to immediately have, unlike what it says at the end of every paper, at the end of every paper, it says there will be a drug within 3 to 5 years, right? Every paper, you probably know this, right? Every paper ends in 2 ways. There will be a drug within 3 to 5 years directed against this discovery, or more research is needed, i.e. fund my lab. That's how they, and so people tend to believe that there's going to be a drug that will just happen as the night follows the day. Unfortunately, that doesn't happen, and this is the whole field of translation. So this does not equal this. And especially when you're a doctor, and neurologists and geneticists are terrible at this. We think that we're done with the left side. So neurologists, as you probably know, are famous for being good diagnosticians. You'll come in and, yes, Mrs. Jones, you can't speak and you can't move your right side. You've had a stroke. Have a nice day. And the patient says, no, wait, hold on a second. What about did you forget the treatment chart? And he's like, oh, yeah. And geneticists are the same way, right? They love giving you a diagnosis. Here's your polymorphism. Here's your mutation. Give it to you with a flourish and have a nice day. And so you could think about the whole area of translation as being not simply understanding why something works, when it works, and when it's broken, why is it how does it break, but how to fix it. And the field of how to fix it is not a field that I got trained in, either scientifically or clinically. How do you develop an intervention that's actually going to improve human health? So that's what translation is. So translation, and the institute at NIH that carries this name, has a bit of a problem because translation has many other terms, many other meanings, right? I mean, no other institute at NIH has this problem. I mean, the National Cancer Institute, everybody knows what cancer is. National Eye Institute, and everybody knows what an eyeball is. National Institute for Advancing Translational Sciences. When I became director of this, I was down in the Congress, and literally I got asked, so you guys do linguistics, right? Is that what you do? It goes just translation, right? Or I had somebody from an NIH grantee who said, you have an entire center at NIH focused on how mRNA gets made into protein? That's a really specific center, because that's translation. So this is translation. How we define it is the whole process by which you go from the moment when a light bulb goes off in the lab, and a person working in the lab, or a doc in the clinic, or in the community. Think about the Ebola crisis just passed recently, where a light bulb goes off and the person says, this observation that nobody has ever made before, I think gives an insight that may lead to an intervention that could improve, it could affect the biology in front of me or the health problem in front of me. The whole process from when that light bulb goes off to creating the intervention, the drug, the device, the diagnostic, whatever it is, showing it's useful in people, and then showing it's useful in public health, in population settings, that you hope it would have when the light bulb went off in the beginning. So that whole process is translation. But you notice that NCAT is not the National Center for Translation. I mean, every institute and center at NIH and probably most organizations in Canada do this, right, as defined. But NCAT is the National Center for Advancing Translational Sciences. So we always like to point out that NCAT is the only institute or center of the 27 at NIH with a verb in the name. right? So advancing. So that tells you that we're really particular about measuring where we are and whether what we're doing is improving the process, the speed, efficiency, cost, et cetera, of the translational process. But it's improving, advancing translational science. So what is translational science? It's the field of investigation that seeks to understand what the general principles are by which this whole process happens. So something that I'll come back to over and over and over again is that the translational process, for a variety of historical reasons, is almost entirely empirical, or in other words, trial and error. And when you have a process that goes from that goes from the beginning to the end, has a dramatic increase in numbers of degrees of freedom as you go forward, the likelihood of your getting to the end of this process via a trial and error process successfully is very small. And so you probably know these numbers. Developing a new drug, just as an example, currently takes about 15 years. Costs, depending on how you do the math, between $1 billion and $10 billion, and fails 99.9% of the time. And the reason for that, there's a lot of reasons, but a lot of the reason for that is we don't understand what the general rules are. And nobody has ever really worked on this problem before NCATs. It's an interesting question. But it's an enormous opportunity. However, this is not just a scientific problem, it's an operational problem. And I'll keep coming back to this. Okay, so this is sort of how we've conceptualized this. We put this on our website. I just want to make a couple of points here. One is that this really is a circular process. It can start with basic research, go to preclinical, clinical implementation in routine practice, and to public health. But it can also go the other way. It can also go the opposite direction. It's not unidirectional. It's bidirectional. And you can make leaps from one of these sections to the other, and there are lots of examples of that. And you notice what's in the center, and we've done this really on purpose, and I'm not going to have time to talk about it much today, but we really have put patients in the center of everything we do for a variety of reasons. And when we do that, we find that the projects are much better focused and actually much more efficient than not having them in the center and involved in the research teams from the very beginning. Okay, so into this opportunity and problem comes NCAT. So what is the NCAT's mission? And I put in red really the kind of bottom line here. It's to generate innovative methods and technologies which improve, which establish the general principles, the general scientific and operational principles by which translation happens, and thereby turn it from being a phenomenological, empirical process into a predictive science. And so the way you have to do this is develop new paradigms, new methods, new ways, new technologies, new ways of doing things that will therefore enhance development, testing, implementation of diagnostics and therapeutics. And so sometimes people ask me, well, okay, so I understand the problem, but what do you guys do? When you come into the lab on Monday morning, what do you guys do? You can't just work on translation. So what are the problems? Well, interestingly, the problems, the roadblocks in the translational process, from a fundamental discovery to public health demonstration of utility of an intervention, have actually been rather well defined. There's lots and lots of papers and studies out there talking about this. There have been many, many fewer papers on how to solve the problems, but there has been quite a bit of analysis, as you might imagine. And the first three are the big reasons that, for instance, drugs fail before they get into people. And the ones in the middle, sort of data interoperability, biomarkers, sort of span the preclinical, clinical spectrum. And all the other ones are in the clinical space. And I want you to notice a couple things about this. First, there's not a disease name in any of these, and that's the point. These are generic problems. I don't care whether you're working on heart disease or toe fungus or cancer, you run into these problems. So they're generic problems in the translational problem. The second is that they fit, as a result of this, what economists would call the tragedy of the commons. And this is really why they haven't been worked on until now. So the tragedy of the commons is this concept that problems which are everybody's problem in general, but nobody's problem in particular, nobody works on. There's a well-established field of economics which studies this, and this has been the case with these problems as well. You know, if you ask yourself, is this a neurological problem that a neurological institute would solve, or a cancer institute would solve, or an ear institute would solve? Well, no, they're generic problems. These are the problems that NCATS works on. They're almost all fit into this tragedy of the commons category. But it's important that they're not just scientific problems, and you all are aware of this because you live in the middle of this issue. That there are organizational issues. The way we do science is in many cases at least non-permissive, if not antithetical, to the requirements of translation. And this has to do with sharing data, for instance, how to handle IP, how do you integrate project management, which is something which is de rigueur in industry, but not in academia, but necessary if you're going to have a team, have team science. How do you give credit in an academic setting for team science, the middle author problem, in other words? How do you give credit for health improvements? I don't know if this is the case in Canada, but at least in the US, for the most part, if you are a student or a postdoc and you make a major improvement in a health system, in the operation of a health system, which results in a tremendous improvement in health outcomes, that is not considered an academic advance on the basis of which you will get academic credit. And that needs to be fixed if translation is going to be a true academic discipline. Education and training, this is both a scientific issue and a cultural issue. By cultural, I mean incorporating non-traditional areas like entrepreneurship, regulatory science, understanding of IP or ethics. And novel public-private partnerships. You know, translation is, by its name, it's a bridge, and so frequently, what you do in this space is you're between traditional academia and traditional commercialization, and so you really need novel models by which to do this. So, before I get into the science, I want to make a point that I put this slide in after our conversation we had last night at dinner. This is a big deal for me, and I think it probably is for you, that translation is a team sport. I don't care how smart you are, I don't care how motivated you are, I don't care how many hours you work, you cannot do this alone. Translation cannot be done alone. It requires top performers from a variety of different fields to work together to a common goal. So I want to contrast this with how I was trained. So I was trained with this guy as my exemplar. So, who is this guy? Black and white photo, holding up a petri dish with crud on it. That's got to be Alexander Fleming, right? I mean, this is the model I had, right? I mean, you're working away late at night by candlelight, and you come in, eureka, you've discovered penicillin. It's a very solitary exercise. All of my original papers were two-author papers, me and the lab, and my lab head. This works fantastically, and I would still argue in many cases, perhaps in most cases, in fundamental research, still works well as the model. But this is science as golf. Right? It's a solitary exercise. Maybe you have a caddy, otherwise known as your lab head, and that's great for fundamental research. But that is not translation. Translation is football, and this is American football. So I think I can use this analogy. I used this analogy in Europe a little while ago, and it completely fell flat. But I think I can do it here. So I just want to take you through this problem here. Okay. So in football, I got to come around here to make the point. Okay, so you probably know in football, the deal is that this guy here, the quarterback, hands off to this running back here, and he's going to try to run around and score, right? But here we have the defense here. This is Mother Nature over here, right? That doesn't want to let her secrets out easily, right? So the only way that this guy is going to score and not get taken out by number 13 here, who's going to try to turn him into hamburger, is number 72 here. And number 72 is going to throw his body in front of number 13 to allow his teammate to score, right? So in football, everybody gets credit, right? So if you win the Super Bowl, everybody gets a Super Bowl ring. Now, imagine this is academia. Okay, so the lab head, who's the quarterback, calls the plays, right? But the person who actually does all the work is the running back. And this guy just sits here and drinks coffee, while the typical lab head, I don't get to say that, it's what I do. And so the running back runs around here. Now, this running back will never be able to score without his teammate endangering his life to allow his teammate to score. Now, if there was a score here, the lab head would be the last author, and the person who scored would be the first author, and number 72 would be a middle author and would not get tenure. So you ask, why don't we have more translational scores in academia? It's because people have to go against their self-interest in order to do it, and we've created a system that perpetuates that. And this is not going to work unless we fix that incentive structure. So with that is my last sports analogy, and now I'm going to get on to some science. So how do we do this? NCATs, this is a huge problem, right? So NCATs cannot solve every problem in all 7,000 human diseases. So everything we do, we have this 3Ds mantra. That is, we develop new ways of getting over one of those roadblocks that I showed you, that long list, in the translational space. Then we demonstrate in individual use cases, a disease or a target or something, that this actually works, and then we actively disseminate it in the whole area of implementation science to demonstrate to others how they can do this. So there's scientific initiatives that NCATS does, not surprisingly. They're divided into clinical, preclinical, and re-engineering, and I'm just going to touch on a few of these programs. I don't have time to go through all of them, of course. So Division of Clinical Innovation, and I realize there's a lot of words on these slides, and I actually did this on purpose because you're going to have these slides, so if you want to look at them later, you can. About 80% of the organization is focused on the problems of translation in the clinical space, so human subjects research. And the goals of this program are training, innovating, communicating, measuring, and partnering. So we spend a lot of time, about a third of the budget goes into training, about a third of it goes in, or more, goes into innovating in getting over some of these roadblocks that I'll talk about in a second in translational science. We find that communication is off-scale important for us because people don't understand. Many people don't even understand that translation exists. And I was testifying in front of the Congress last week, and this was brought home to me, that many people don't appreciate that this problem even exists. But then we have to measure whether we're successful, right? Or else we don't know whether we're advancing translational science. And again, translation is a team sport. So everything NCATS does is a collaboration with somebody, some organization or collaborator somewhere in the world. So most of this goes through a program called the Clinical and Translational Science Award Program. It's the NIH's biggest single program. It supports a national consortium of medical research institutions like UT in the U.S., improves the way clinical and translational science is done, and provides innovative training. These are the spots. If you picture your favorite academic institution in the U.S., most of them have these. There's 62 of them now, and the way this organization worked until NCATS got formed is that each one worked as a silo. They did almost nothing together, with some rare exceptions. And what we're doing with this program is to tie all 62 of these together into a true national network for translational medicine that has a common electronic health record structure so we can recruit patients across the 62 centers, a common IRB structure, common clinical trial, clinical site qualification process that then will focus on innovative ways of doing clinical trials, adaptive trials, things like that. So a couple of things that this program is doing. So first is training the translational research workforce. And this workforce requires different skills and attitudes than at least the way I was trained. So non-traditional areas such as regulatory science or entrepreneurship. We're big on externships in industry, foundations, FDA, et cetera. The reason we do this is absolutely straightforward. If you think about the word translation, translation means literally to carry across. Most of you took foreign language in high school or college, university. When you did that, you probably had an experience, if you got into learning a foreign language, where you went to the home country. You went and did a study abroad. And you learned the word use, the vernacular, the history, the culture, and you could begin to look on problems and issues from their point of view, not your point of view, so that you could be a good translator. This is exactly the same issue. We can't train people in an academic setting about what it's like to be in a company and expect them to know what it's like to be in a company. They have to go work in a company or at the FDA or anywhere else. And so having a training system that does this is really important. Fostering team science, the way we were talking about. And this, it's interesting, this last thing, it's interesting. Creating an environment where translational research is a viable career path. I'm sure that is of interest to all of you. In many places in the U.S., though there's a lot of talk about this, promotion and tenure systems have not caught up with this. And so this is something that's a real issue that we're working on very hard. Another thing we're doing is streamlining multi-site clinical studies. You probably know that the current system is incredibly inefficient. There was a paper published by some folks at the Oregon Health Sciences University about 3 or 4 years ago that showed that at their institution, 75% of NIH-funded trials fail to recruit. 75%. That means that all this money goes into a clinical trial and nothing comes out of it. It's just, if this was a lung, this would be dead space ventilation. It's amazing, and it has nothing to do with the science. It has to do with the operations of how this works. This is a solvable problem, but nobody's ever worked on this before. So centralized IRB review, centralized contracting, things which, is that going to give you on the cover of Cell? No. But is this going to get more patients, more treatments to more patients more quickly? Absolutely. And is it going to create a platform on which we can do really exciting, innovative clinical research? So we're creating these things called trial innovation centers that'll provide a central institutional review board, streamlined budgeting, contracting, coordination functions, and these will partner with NIHIC institutes and centers, industry, et cetera, to implement trials. Improving efficiency via participant recruitment. I just gave you this rather sobering number of the number of times that a clinical trial fails. I did a lot of mouse work earlier in my career, and I can tell you I never had a mouse experiment fail for lack of recruitment. Right? But this is a problem. It is a major, major problem in human studies. It's very clear that if there were drugs for every one of those untreatable diseases tomorrow, we would not have a system to test whether those interventions are safe and effective. And this is a solvable problem. This is not, in this day and age, this is not a technical problem. It is a cultural problem and an organizational problem. And so we're putting in place what are called recruitment innovation centers that are going to provide data for trial planning, feasibility, analysis, site selection, using a common electronic health record structure that allows you to qualify patients for clinical trials without going, all the data can be anonymized, so you don't have to worry about HIPAA or IRBs, at least initially. And then once the trial is funded, then use that infrastructure to be able to do the recruitment in a very rapid fashion. So the idea here is that within a year or two, we're about halfway through this now, that the CTSA program will really transform the way clinical translational science is done by having these 62 hubs, which then have these innovation centers to do trial operation and recruitment. And then there'll be multi-site studies that are funded by others, NIH or others. And the analogy we're using here is that this network will be like a computer into which you can hook your USB port if you have say a new therapeutic or a new device or something that you want to study. And you don't need to rebuild the components every time. I don't know how this works in Canada, but in the U.S., a huge amount of money is spent every time a clinical trial is done to create all the infrastructure to do that trial. Then the trial goes on for 5 or 6 years, and all the infrastructure is torn down again. And this would be like if you knew that you needed a house, every day when you left, you'd tear down the house, and then you'd come back after work and have to build the house again to live in it. And if somebody did that, you'd say, why don't you just build a house that a lot of things can be done in? And that's the kind of situation that we're in, believe it or not, in this day and age. So solvable problem. Okay, secondly, if I'm going to work backwards here from the clinical to the preclinical stage. So we do a lot of work on rare diseases. Why? A couple of reasons. First of all, the majority of those untreatable diseases are rare diseases, defined in the U.S. as less than 200,000 patients. Secondly, they tend to be multi-organ system disorders. They're the original precision medicine disorders. That is, they're defined by genetics, generally in this day and age, not by symptomatology. Not by organ pathology. And so they don't fit neatly into an individual center or institute structure the way NIH has, so they tend to be neglected. Secondly, if you're trying to innovate in clinical, in translational processes, you want to do demonstration projects in relatively simple systems. And rare diseases are sort of the model organisms of the translational space, if you want to think of it that way. And if you're going to try something out that's novel, a novel recruitment strategy or a novel way to do toxicology, et cetera, you don't want to do it with a very complex disorder. You don't want to do that studying autism. You want to do it studying Menkes disease, which is a single gene disorder of copper metabolism, as an example. So we have, and this is the Big Bang diagram of this organization, we have another network called the Rare Disease Clinical Research Network, and you can see the names around here that has two important distinctions to it. First of all, each one has what's called a PAG around here, and a PAG stands for Patient Advocacy Group. So in order to get funded, you have to have a patient group as part of your research team, or else you don't get funded. Really important, and this has been essential to the efficiency of this program. Secondly, you don't just work on a single disease. So the idea that each one of these 6,000 rare diseases is an independent observation is simply inconsistent with what we know about biology. Biology is connected. These disorders are all connected in one way or another. And so each of these networks, each of these consortia has to look at commonalities. So they have to ask, well, either there's a phenotype like brittle bone disorders, or it's an organelle, like I think up here there's a mitochondria or a lysosome, or it's a biochemical pathway like urea cycles, et cetera. But then they study 10, 20, 30 of these disorders at one time, looking at their commonalities to try to hit more birds with one stone, if you will. Another program which I just want to mention briefly, which addresses a different problem, is this new therapeutic uses program. So again, I'm moving backwards into the preclinical space now. So this addresses the translational problem that sort of reciprocal of what happens in drug development. So remember I said that in drug development about 99% of projects fail from beginning to end. Even when a drug gets into people, somewhere between 80 and 90% of those drugs that are tested at first in people are never approved for any use. And sometimes they fail for toxicity, but more often they fail for efficacy. They don't work in the indication that they were designed for, or they're deprioritized by the company for business reasons. And so it was evident to a number of years ago that, well, gosh, for every FDA-approved drug, there's 4 or 5 or even 9 partially developed drugs to which these companies have put huge amounts of effort, hundreds of millions of dollars sometimes, and they're not benefiting anybody. And they're not benefiting the company either because they're not able to sell them to do anything. So what if we hooked those assets, those partially developed drugs together with academics who had novel ideas to which the drugs that affect these pathways could be put? Then maybe we could develop new therapeutics in a very rapid fashion. And so what we did was to create a couple of things which were catalysts, again, to make this happen. We created a platform by which the pharmas, the 8 pharmaceutical companies that offered their 60 compounds, and academics could find each other. And this is essentially a Match.com sort of platform that we created so that people could find each other in cyberspace. Secondly, we created novel template documents to get around the lawyers. So I hope there's no lawyers here, but you probably know, if you've done these kinds of collaborations before, that working out a collaborative agreement between an academic and a company can take a year, 2 years sometimes. And so we drew up a template agreement, gave everybody 11 weeks to negotiate their agreement, and all but one of them did it. So again, it's not a strictly scientific issue, but an issue which dramatically sped up the process by which these were tested in people. And of course, we provided money as the ultimate catalyst. And I want to show you, this is the first round of the projects that were funded last year. So if you look at the diseases, they're quite broad in their indication space. They are all either untreatable or poorly treatable, and they're 2 rare diseases, and the rest are common. And there's an academic partner and a pharma partner, and these really are tight collaborations between the two. And we're measuring these. Remember, we measure everything. We're measuring whether the use of a template agreement speed negotiation time. They certainly do. Whether crowdsourcing of our indications gives us new indications, new ideas that nobody would have thought of. That certainly is the case too. And we're just beginning to identify whether any of these studies result in new indications. And I just want to give you a kind of a tantalizing finding that came out just about 3 weeks ago. So this was a very interesting story of an AstraZeneca drug that is a Src kinase inhibitor that was developed for cancer and was deprioritized by AstraZeneca because it didn't have, at least as a single agent, it didn't have the kind of therapeutic efficacy that they were looking for. So they put it on the shelf. Well, it turns out that one of the Src kinase families is something called FYN, F-Y-N. And an investigator, Steve Strittmatter at Yale, who works on Alzheimer's disease, discovered that FYN kinase is involved in the process by which A-beta oligomers induce neuronal synaptic dysfunction, which is one of the first defects in Alzheimer's disease. So the concept was, by blocking FYN kinase, you're not going to block formation of amyloid oligomers, but what you might do is prevent the neuronal damage that those oligomers cause. Really interesting idea. And in this animal model that just got, it's a double transgenic, APP-PS1 transgenic in Alzheimer's, this drug not only reverses the behavioral deficits in these animals, but it also reverses the synaptic pathology in these animals. And given the way that, given how much was known about this drug, we actually did the phase 1 trial at the same time as we did the animal study, and so we're actually into a phase 2A trial within 18 months of funding this. So normally this would have taken 10 years, and now we've done this in 18 months. And so this can happen. It can happen very fast, but you have to think about novel ways of doing things, question everything you do, look at what the limiting reagents are like you would in a biosynthetic pathway, and attack those individually with a catalyst to make it happen faster. Okay, so now I'm going to move farther back. Let's say you have a target and you don't know what to do with that target. That's where the NCAT's Chemical Genomics Center comes in. This is a collaborative program where there's about 100 people at NCAT's who have deep drug development expertise. Most of them come from pharma and biotech that work with academic investigators who have targets that they discover that they think might be important for a disease, but they don't know how to get started developing a proto-drug that might be useful for them. And that's what this organization does. Assay development, high-throughput screening, medicinal chemistry, sort of target to lead, if you want to think of it that way. And the focus is on unprecedented diseases, novel targets. So here's just an example. And this is, I could show you, 50 different examples like this, but I like this one for a variety of reasons. So here's an enzyme, a human enzyme called 12-lipoxygenase. So it's been known for about 30 years, and it's involved in the arachidonic acid pathway. And I'm not going to go into what it does, but it's involved in inflammation, as you might imagine. And 12-HLO has been known for over 30 years, but because of the lack of specific pharmacological inhibitors, it's been impossible to figure out what this enzyme actually does. And so we collaborated with an investigator at UC Santa Cruz, Ted Holman, who's a biochemist, who wouldn't know a drug if it hit him in the head, and I wouldn't know a patient if the patient hit him in the head, but that's okay, because our folks didn't know how to spell lipoxygenase before we started working with Ted Holman. So we formed a joint project team that developed developed an assay, developed the screen, did the screen of hundreds of thousands of compounds, did medicinal chemistry optimization, led to this first specific inhibitor, and allowed us to figure out in vivo what this enzyme does. Well, it turns out when you do that it's involved in platelet aggregation and in the inflammation that happens early in the process of type 1 diabetes. That then led to other collaborations, a guy named Jerry Nadler in Virginia who works on diabetes, and Mike Hollenstadt at Jefferson who works on platelet aggregation. And then that led to an agreement with Sanofi to develop these further. So this is a classic example of de-risking a target, having a drug go from the undruggable to the druggable category. And led to lots of publications, which resulted in Ted Holman getting a promotion at UC Santa Cruz, which is a good thing as well. Okay, so let's say you have a proto-drug like that, and now you want to go into people. So then what do you do? Well, you got to have a preclinical development program that's going to take you from a lead, like what I just showed you for 12-HLO, to something which can be tested in people. So again, this same model, collaboration between NCATs and external investigators who have a lead or an asset that they want to develop that can be tested in people. You can get into this program anywhere from a probe, like what I just showed you, to something which needs what are called IND enabling work to be able to get an investigational new drug application to put a drug in people from the FDA. And the whole point of this is to take each of these projects to the point where it's de-risked enough that a company would adopt it and take it through the rest of clinical development. So de-risking, that's the whole idea. And this is what the current portfolio is. The only reason I show you this is, again, to show you how diverse the target indications are. So these are all rare diseases, and you probably haven't heard of most of these, but they go from pulmonary diseases to intellectual disability disorders, to cancer, to endocrine disorders, to infectious disorders, neurodegenerative diseases. About half of them are collaborations with academics, about half of them are collaborations with small companies. And I'm just going to give you one example. This is a project on sickle cell disease. So I mentioned sickle cell disease before, and one other thing I want to show you after this, but I'm coming back to where I started. So this collaboration on sickle cell disease, it's an interesting object lesson, and it's a good endpoint too. So that's always a happy story. So there was this drug, a potential drug up here, called S103. It was actually discovered by an academic at Virginia Commonwealth University about 20 years ago, but he was a basic researcher like Ted Holman at Santa Cruz and really didn't know what to do with it, and he retired, and the asset was available, and the CEO of this company, SRX, was a little company in Boston, identified this and as a potential treatment for sickle cell disease, but he was unable to get any funding to move this thing forward, even though it's a tremendous unmet medical need. It's about 100,000 people in the U.S. that have this disorder. It's a terribly morbid, it's a disorder with much morbidity and mortality, and frankly, it's embarrassment, I think, from the standpoint of being able to develop treatments based on genetic knowledge. And so why wasn't this movable forward? And this is a general thing that you run into over and over again. First thing is, this compound is really scary. Now, chemists are easily scared, I grant you that. But from a chemist standpoint, just take my word for it, that is a scary structure. It has lots of scary potential features to it. It's very reactive. So that's first risk. Second risk is, even though it had a mechanism of action, which is very exciting, it was potentially also a real problem, target risk. So what this compound does is it binds covalently to sickle hemoglobin and shifts the oxygen dissociation curve to the left so that the sickle erythrocytes can get through the hypoxic environment of the postcapillary venules without sickling. And so it directly reverses the molecular defect that Linus Pauling discovered. That's very exciting. However, if this compound covalently binds to sickle hemoglobin, it might covalently bind to every other protein in the proteome and cause all sorts of toxicity. It doesn't, but that was the concern. So chemical risk, target risk. Clinical risk. Clinical studies in this population are really hard for a variety of reasons. There's a lot of, the main symptom of this disorder is pain. So many of these patients are on chronic pain meds, and it's a difficult population as a result to do a clinical study in. So nobody wanted to approach it for that reason. And the fourth reason was regulatory risk. So the FDA has a rather checkered history of reviewing a sickle cell program. So chemical risk, target risk, clinical risk, regulatory risk. No organization would fund this. We, however, looked at this and we said, "You got to be kidding me! This is a remarkable opportunity." So we partnered with SRX. In less than a year, we were in people. Did all the toxicity, all what's called the CMC studies, all the regulatory studies. We're in people. Did a phase 1, 1A, 1B, 2A study, and on the basis of the efficacy shown in that the initial efficacy and complete safety of this compound in sickle cell patients, it was acquired. The whole project was acquired by Baxter International for completion of the clinical development, and they're now doing the definitive clinical trials. So again, this can be done, and it can be done rapidly, but you have to think differently. You have to think of this as a team sport and approach it that way. And this suggests that what we have in drug development is actually not an immutable law. So, what is that law? So you all know what Moore's law is, I hope. If you don't know what Moore's law is, don't admit it. Google it afterwards. Moore's law is why you could fit a mainframe computer when I was in college into one of these things. It's the increase in number of transistors you can fit on a microprocessor, which has increased linearly over the last 40 years, named after Gordon Moore, who's one of the co-founders of Intel. And in drug development, we have what's called Eroom's law. Now, this is Moore's law spelled backwards. This comes from a real paper that was published a couple of years ago, and this is a terrifying graph, and it should terrify all of you, and it terrifies all of us who are within shooting range of Alzheimer's disease, I must tell you. So what are you looking at here? This is the opposite of Moore's law. So this is dramatically negative productivity growth in the pharmaceutical, in the development of drugs since 1950. So what you're looking at here is that the number of new drugs that were approved by FDA per $1 billion spent corporately by the whole research enterprise has gone down by half every 9 years since 1950. So just imagine if you had any other industry where their efficiency of creating whatever they create goes down linearly over 60 years, every one of those companies would go out of business. And that's what is going on with pharma companies, and that's in part why drugs are so expensive now. And if you ask, where does this go to zero? Formally, it goes to zero in 2045. So formally, there will be no more drugs developed after 2045, or in other words, a new drug will be infinity, cost infinity, which is pretty much the same thing. And what's terrifying about this is that think about the epochal changes in science and medicine that have happened since 1950. Invention of computers, invention of recombinant DNA, you can imagine, on and on and on. None of them have caused a change in this line. This is one of the reasons that NCAT was formed, and it ought to be terrifying to all of us. So is this bendable? And I would argue to you that the early evidence is that it is bendable, and I don't have time to go into this, but it's a paper that we published in Science Translational Medicine in January, together with a finance professor at MIT named Andrew Lowe, who took all the data from our rare disease portfolio and plotted likelihood of success and cost, comparing it to industry averages. And without going into the details, our success rates are considerably higher, and the costs are considerably lower than industry standard. And I don't have time to get into why we think this is, but the point is that I do think that Eroom's law is bendable. It's not an immutable law of nature, but it requires you to think different. If you keep doing the same stuff, you're going to get the same outcome. Okay, so lastly, a real challenge for you, if I haven't given you enough of them so far. So this is a more basic challenge. So toxicity is a major reason that drugs fail during development. About 30% of the time, when you're trying to develop a new drug, it fails either preclinically in animals or clinically in people because of an unanticipated adverse event. And so the problem with this was that the way toxicity is assessed has not changed in the last 50 years. So the way toxicity is assessed essentially is you give a dose to an animal, and then you shut your eyes and you look at what's called an apical endpoint at the other end. That is, the animal dies, develops liver failure, develops a cancer, but you really have no idea why. So the next time you have a drug, you just have the same black box system. So a number of years ago, we thought, well, why don't we try to dissect this process using a screening of various sorts of targets and pathways and cells, using high throughput screening, high content screening, and computational models to model why drugs and chemicals are actually toxic. And so we formed a collaboration. This will not surprise you now, a novel team between 4 government agencies. This is the FDA, the EPA, the National Toxicology Program, and ourselves. And the purpose of this program is nothing less than to characterize response of 10,000 chemicals in every pathway operant in mammalian cells. So this is the grandmother of all systems biology experiments, with the hypothesis that any pathway that's involved in physiology, if you mess it up, could lead to pathophysiology, otherwise known as toxicity. So we have to understand every pathway that's interdicted by any of these chemicals as a way to predict whether toxicity is going to happen in people. And this currently has about 10,000 chemicals in it. We contributed ourselves every drug that's ever been approved for human use worldwide, and a lot of investigational drugs we have in our collection. The EPA and the NTP contributed not only human use chemicals, but pesticides, other environmental chemicals into this 10,000 set. And I don't want to go through this in any detail, but just to show you that what happens is that every one of these assays goes through a really neurotic process of robotic validation, just throw a number at you. Each one of these 10,000 compounds is run in triplicate 15-point concentration response on a different assay every week. So we generate about 600,000 data points every week, and all those data are put in the public domain for you to model. And so there's a lot of data sharing and then goes into the public domain. And just as an example, we've spent a lot of time working on nuclear hormone receptors, agonists and antagonists of estrogen receptor signaling pathway, which is a big deal in environmental and drug-related toxicity. Believe it or not, there really was no data in the public domain that addressed this problem up until we did this study. And so we're now working on about 100 other pathways, and all of those data will be made public. So another approach to this problem, which I just want to leave you with, is this tissue chip program. So as opposed to the Tox21 program, which is an unabashedly reductionist approach to the toxicity problem, is a different approach to say, well, if the problem is that animal testing is not necessarily predictive of human toxicity, why can't we develop little human organoids in microfluidic platforms that would model a structure and function of human organs in a way that would allow these little organoids to be used to test for safety and efficacy. I must say, when we started doing this 3 years ago, even I was a skeptic. I really thought this was a bridge too far. I'm glad to tell you that it isn't. This project is ahead of its milestones in every way. So what this program is trying to do is to model 10 different organs, and you can see them listed here, in a physiologically relevant, anatomically accurate way, in a modular, configurable platform, so that all of these are being designed kind of like Lego bricks, so you can mix and match them. They have to be viable for at least 4 weeks without pen strep, and those of you who've done tissue culture know how challenging that is. And they have to be accessible to the broader community. So here's just an example of how you do this. For any one of these organs, you have to start out with a scaffold, generally extracellular matrix. Then you have to add cells, which are usually iPS cells, or sometimes they're primary cells. They have to have the correct structure. Sometimes the cells will take on that structure themselves. Sometimes you have to give them the structure that they adapt to. They have to have the right spatial and temporal patterning. They sometimes have to be perfused, because remember that these have to be alive for 4 weeks or more. They have to be in a bioreactor, which will keep them alive for this amount of time. Depending on the effect that you're looking for, sometimes they have to be innervated. For instance, we have a gut platform that now is innervated with enteric neurons in the gut, actually peristalsis on the chip. has to have a host response frequently, because host response is often very important to toxicity. You got to have a functional readout. You have to have some sort of biosensor, and then you got to have a computational design that's going to gather all of this. So here's just an example. This is one of my favorite examples because it's easy to kind of picture this. So this is if you're developing a potential heart system. So what you do is you're making heart brownies here. You take iPS cells and you put them in essentially a bakery pan here, and they spread out kind of like brownies. Then you can cut out the little brownies here. And what's interesting is if you leave the brownies, the cardiomyocyte brownies in the dish, they don't look or act or respond like adult cardiomyocytes. They will beat, but they're immature. The only way to get them to respond normally, the way a normal heart would to drugs, is to take the brownie out, attach it to the rack here that then stretches them like a normal heart would. So there's systole, diastole, and when you do that, the cells terminally differentiate and begin to respond to drugs in the way that a heart in situ would. And we find this over and over and over again as a theme with these organs. So then what can you do with this? Well, here's just one early example. We had designed this as a way to do toxicity testing of potential drugs, but very early on, investigators, this happened to be an investigator at Children's Hospital in Boston, thought, well, gosh, why can't I use this to model a rare disease? So this is Bart's syndrome is a rare disease, as a typical multi-system disease, but these kids die of a cardiomyopathy, it's a dilated cardiomyopathy. So the investigator said, well, why don't I try to make one of these heart chips out of Bart's syndrome iPS cells? So that's what he did. So here's the heart on a chip BART model. Here's what a normal heart chip looks like. This is a BART syndrome heart model. This is a rescue. This is a mutation in a gene called tafazin-1. So if you transfect in tafazin-1, it restores the normal architecture. And the important thing is, if you look at the chip down here, this one is a normal chip, normal iPS cell chip. This is a chip made with BART syndrome. iPS cells from Bart syndrome kids, and you notice that there's a little bit of contractility, but not very much. However, if you rescue with wild-type tafazin, you restore normal contractility of these cardiomyocytes. It's a remarkable model of Bart syndrome cardiomyopathy, and this actually was published last year, and it was named the top scientific advance by the AHA last year. So the model that we have here, the endpoint that we have here, that would have seemed really too much Star Trek or Star Wars even 5 or 10 years ago, we're really close to realizing, at least as a research tool. And the reason is important to remember before I tell you what the vision here is. Why did this work? It's because of what we see in translation over and over and over again. That is this concept of convergence. So this is possible because of the convergence of 4 different scientific developments: the development of iPS technology, biosensor technology, tissue printing technology, and microfluidic technology. One of them alone wouldn't have done this, but all 4 of them have allowed this kind of technology to happen. So eventually, what we hope to have is within the next 5 years to have all of these 10 organs, and we're in the process of doing this, all these 10 organs on a single microfluidic platform where, for instance, if you have a novel drug, you could put this novel drug in through artificial blood here to the artificial intestine, and it gets absorbed or not, goes to the artificial liver, gets metabolized or not, goes to the artificial kidney and shows tubular necrosis or not, etc. And you can put in or take out sample compounds at any part of this process, and so you can have all of these readouts here, which then need to be correlated with in vivo endpoints. And the important thing is that we want this not only as a better way of testing potential drugs, but as a potential way to in regulatory settings to avoid doing animal testing for FDA, and FDA is involved in this from the beginning. So once this is done, imagine what one could do. These are iPS cells, so we could make a chip out of every one of you. Right? You could have your own chip. And then one could imagine doing pharmacogenetics on a chip, doing a clinical trial on a chip. And we're already envisioning doing this, running a clinical trial in parallel in people and on chips made from those people. And if I think back to when I was in training 30 years ago, what I just said, without getting taken off to the Mass Mental Health Center and locked up, I would tell you no. This is reality. And so for you students out there, I know you're tired of hearing this, and I know you're tired of hearing this because it's a poor funding environment and all that stuff, but my heavens, the opportunity and the need are so great, and there are such exciting opportunities out there. I just can't tell you what a wonderful time it is for you to be getting into this field, and anything that I can do and your mentors can do to help you along that process, we are more than willing to do and help you to do. So I want to have you to have, in case you want information about any of these programs, I'll give you the program leads. These are all real people at NCATs with real email addresses. And if you want to learn more about us, we've got all the usual sources of information. So thank you for having me. I'm going to be here for the rest of the day, so I'll be glad to talk to any of you during lunch or afterwards. So thank you.